Photo shows a white Arctic fox that blends in with the snow. Figure 33. An Arctic fox is a complex animal well adapted to its environment. It changes coat color with the seasons and has longer fur in winter to trap heat. Credit modification of work by Keith Morehouse, USFWS. Chapter outline. 33. 1 Animal form and function. 33. 2 Animal primary tissues. 33. 3 Homeostasis. The Arctic fox is an example of a complex animal that has adapted to its environment and illustrates the relationships between an animal's form and function. The structures of animals consist of primary tissues that make up more complex organs and organ systems. Homeostasis allows an animal to maintain a balance between its internal and external environments. Learning objectives. By the end of this section, you will be able to do the following. Describe the various types of body plans that occur in animals. Describe limits on animal size and shape. Relate bioenergetics to body size, levels of activity, and the environment. Animals vary in form and function. From a sponge to a worm to a goat, an organism has a distinct body plan that limits its size and shape. Animals' bodies are also designed to interact with their environments, whether in the deep sea, a rainforest canopy, or the desert. Therefore, a large amount of information about the structure of an organism's body, anatomy, and the function of its cells, tissues, and organs' physiology can be learned by studying that organism's environment. Body plans. Illustration A shows an asymmetrical sponge with a tube-like body and a growth off to one side. Illustration B shows a sea anemone with a tube-like, radially symmetrical body. Tentacles grow from the top of the tube. 3 vertical planes arranged 120 degrees apart dissect the body. The half of the body on one side of each plane is a mirror image of the body on the other side. Illustration C shows a goat with a bilaterally symmetrical body. A plane runs from front to back through the middle of the goat, dissecting the body into left and right halves, which are mirror images of each other. The top part of the goat is defined as dorsal, and the bottom part is defined as ventral. The front of the goat is defined as anterior, and the back is defined as posterior. Figure 33. 2 animals exhibit different types of body symmetry. The sponge is asymmetrical, the sea anemone has radial symmetry, and the goat has bilateral symmetry. Animal body plans follow set patterns related to symmetry. They are asymmetrical, radial, or bilateral in form as illustrated in Figure 33. 2 asymmetrical animals are animals with no pattern or symmetry. An example of an asymmetrical animal is a sponge. Radial symmetry, as illustrated in Figure 33.2, describes when an animal has an up and down orientation. Any plane cut along its longitudinal axis through the organism produces equal halves, but not a definite right or left side. This plan is found mostly in aquatic animals, especially organisms that attach themselves to a base, like a rock or a boat, and extract their food from the surrounding water as it flows around the organism. Bilateral symmetry is illustrated in the same figure by a goat. The goat also has an upper and lower component to it, but a plane cut from front to back separates the animal into definite right and left sides. Additional terms used when describing positions in the body are anterior, front, posterior, rear, dorsal toward the back, and ventral toward the stomach. Bilateral symmetry is found in both land-based and aquatic animals. It enables a high level of mobility, limits on animal size and shape. Animals with bilateral symmetry that live in water tend to have a fusiform shape. This is a tubular-shaped body that is tapered at both ends. This shape decreases the drag on the body as it moves through water and allows the animal to swim at high speeds. Table 33. 1 lists the maximum speed of various animals. Certain types of sharks can swim at 50 kilometers per hour, and some dolphins at 32 to 40 kilometers per hour. Land animals frequently travel faster, although the tortoise and snail are significantly slower than cheetahs. Another difference in the adaptations of aquatic and land-dwelling organisms is that aquatic organisms are constrained in shape by the forces of drag in the water, since water has higher viscosity than air. On the other hand, land-dwelling organisms are constrained mainly by gravity, and drag is relatively unimportant. For example, Most adaptations in birds are for gravity, not for drag. Maximum speed of assorted land and marine animals. Animal speed, kmh speed mph. Cheetah 113.70. Quarter horse 77.48. Fox 68.42. Shortfin mako shark 50.31. Domestic house cat 48.30. Human 40.528. Dolphin 32, 40.20.25. Mouse 13.8. Snail 0, 0.50, 3. Table 33, 1. Most animals have an exoskeleton, including insects, spiders, scorpions, horseshoe crabs, centipedes, and crustaceans. Scientists estimate that, of insects alone, there are over 30 million species on our planet. The exoskeleton is a hard covering or shell that provides benefits to the animal, such as protection against damage from predators and from water loss for land animals. It also provides for the attachments of muscles. As the tough and resistant outer cover of an arthropod, the exoskeleton may be constructed of a tough polymer, such as chitin, and is often biomineralized with materials such as calcium carbonate. This is fused to the animal's epidermis. Ingrowths of the exoskeleton, called apodemes, function as attachment sites for muscles, similar to tendons in more advanced animals, figure 33.3. In order to grow, the animal must first synthesize a new exoskeleton underneath the old one, and then shed or molt the original covering. This limits the animal's ability to grow continually, and may limit the individual's ability to mature if molting does not occur at the proper time. The thickness of the exoskeleton must be increased significantly to accommodate any increase in weight. It is estimated that a doubling of body size increases body weight by a factor of 8. The increasing thickness of the chitin necessary to support this weight limits most animals with an exoskeleton to a relatively small size. The same principles apply to endoskeletons, but they are more efficient because muscles are attached on the outside, making it easier to compensate for increased mass. Illustration shows a crab claw with a small upper portion that pivots relative to a large lower portion. The apodemes are located on the large portion, above and below the pivot point. Figure 33. 3 apodemes are ingrowths on arthropod exoskeletons to which muscles attach. The apodemes on this crab leg are located above and below the fulcrum of the claw. Contraction of muscles attached to the apodemes pulls the claw closed. An animal with an endoskeleton has its size determined by the amount of skeletal system it needs in order to support the other tissues, and the amount of muscle it needs for movement. As the body size increases, both bone and muscle mass increase. The speed achievable by the animal is a balance between its overall size and the bone and muscle that provide support and movement. Limiting effects of diffusion on size and development. The exchange of nutrients and wastes between a cell and its watery environment occurs through the process of diffusion. All living cells are bathed in liquid, whether they are in a single-celled organism or a multicellular one. Diffusion is effective over a specific distance and limits the size that an individual cell can attain. If a cell is a single-celled microorganism, such as an amoeba, it can satisfy all of its nutrient and waste needs through diffusion. If the cell is too large, then diffusion is ineffective, and the center of the cell does not receive adequate nutrients, nor is it able to effectively dispel its waste. An important concept in understanding how efficient diffusion is as a means of transport is the surface-to-volume ratio. Recall that any three-dimensional object has a surface area and volume. The ratio of these two quantities is the surface-to-volume ratio. Consider a cell shaped like a perfect sphere. It has a surface area of 4 pi/2 and a volume of 4/3 pi/3. The surface-to-volume ratio of a sphere is 3R. As the cell gets bigger, its surface-to-volume ratio decreases, making diffusion less efficient. The larger the size of the sphere or animal, the less surface area for diffusion it possesses. The solution to producing larger organisms is for them to become multicellular. Specialization occurs in complex organisms, allowing cells to become more efficient at doing fewer tasks. For example, circulatory systems bring nutrients and remove waste, while respiratory systems provide oxygen for the cells and remove carbon dioxide from them. Other organ systems have developed further specialization of cells and tissues and efficiently control body functions. Moreover, surface-to-volume ratio applies to other areas of animal development, such as the relationship between muscle mass and cross-sectional surface area in supporting skeletons, and in the relationship between muscle mass and the generation and dissipation of heat. Link to learning. Visit this site to see a time-lapse video of zebrafish development. Animal bioenergetics. All animals must obtain their energy from food they ingest or absorb. These nutrients are converted to adenosine triphosphate, ATP, for short-term storage and use by all cells. Some animals store energy for slightly longer times as glycogen, and others store energy for much longer times in the form of triglycerides housed in specialized adipose tissues. No energy system is 100% efficient, and an animal's metabolism produces waste energy in the form of heat. If an animal can conserve that heat and maintain a relatively constant body temperature, it is classified as a warm-blooded animal and called an endotherm. The insulation used to conserve the body heat comes in the forms of fur, fat, or feathers. The absence of insulation in ectothermic animals increases their dependence on the environment for body heat. The amount of energy expended by an animal over a specific time is called its metabolic rate. The rate is measured variously in joules, calories, or kilocalories, 1,000 calories. Carbohydrates and proteins contain about 4.5 to 5 kcal-G, and fat contains about 9 kcal. G. Metabolic rate is estimated as the basal metabolic rate, BMR, in endothermic animals at rest, and as the standard metabolic rate, SMR, in ectotherms. Human males have a BMR of 1,600 to 1,800 kcal-day, and human females have a BMR of 1,300 to 1,500 kcal-day. Even with insulation, endothermal animals require extensive amounts of energy to maintain a constant body temperature. An ectotherm, such as an alligator, has an SMR of 60 kcal-day. Energy requirements related to body size. Smaller endothermic animals have a greater surface area for their mass than larger ones, figure 33.4. Therefore, smaller animals lose heat at a faster rate than larger animals and require more energy to maintain a constant internal temperature. This results in a smaller endothermic animal having a higher BMR per body weight than a larger endothermic animal. A mouse has an average mass of 35 grams and a metabolic rate of 890 millimeters cubed of oxygen per gram body mass per hour. The elephant has an average mass of 4,500 kg and a metabolic rate of 75 millimeters cubed of oxygen per gram body mass per hour. Figure 33. The mouse has a much higher metabolic rate than the elephant. Credit mouse, modification of work by Magnus Kjaergaard. Credit elephant, modification of work by the lizard queen/flicker. Energy requirements related to levels of activity. The more active an animal is, the more energy is needed to maintain that activity, and the higher its BMR or SMR. The average daily rate of energy consumption is about 2 to 4 times an animal's BMR or SMR. Humans are more sedentary than most animals and have an average daily rate of only 1.5 times the BMR. The diet of an endothermic animal is determined by its BMR. For example, the type of grasses, leaves, or shrubs that an herbivore eats affects the number of calories that it takes in. The relative caloric content of herbivore foods in descending order is tall grasses, legumes, short grasses, forbs, any broad-leaved plant, not a grass, subshrubs, annuals, biennials. Energy requirements related to environment. Animals adapt to extremes of temperature or food availability through torpor. Torpor is a process that leads to a decrease in activity and metabolism and allows animals to survive adverse conditions. Torpor can be used by animals for long periods, such as entering a state of hibernation during the winter months, in which case it enables them to maintain a reduced body temperature. During hibernation, ground squirrels can achieve an abdominal temperature of 0 degrees C, 32 degrees F, while a bear's internal temperature is maintained higher at about 37 degrees C, 99 degrees F. If torpor occurs during the summer months with high temperatures and little water, it is called estivation. Some desert animals use this to survive the harshest months of the year. Torpor can occur on a daily basis. This is seen in bats and hummingbirds. While endothermy is limited in smaller animals by surface to volume ratio, some organisms can be smaller and still be endotherms because they employ daily torpor during the part of the day that is coldest. This allows them to conserve energy during the colder parts of the day when they consume more energy to maintain their body temperature. Animal body planes and cavities. A standing vertebrate animal can be divided by several planes. A sagittal plane divides the body into right and left portions. A midsagittal plane divides the body exactly in the middle, making two equal right and left halves. A frontal plane, also called a coronal plane, separates the front from the back. A transverse plane, or horizontal plane, divides the animal into upper and lower portions. This is sometimes called a cross-section, and if the transverse cut is at an angle, it is called an oblique plane. Figure 33. 5 illustrates these planes on a goat, a four-legged animal, and a human being. Illustration A shows the planes of a goat body. The midsagittal plane runs through the middle of the goat from front to back, separating the right and left sides. The frontal plane also runs from front to back, but separates the upper half of the body from the lower half. The transverse plane runs across the middle of the goat and separate the front and back halves of the body. Illustration B shows the planes of a human body. The midsagittal plane runs from top to bottom and separates the right and left halves of the body. The frontal plane also runs from top to bottom and separates the front and back halves of the body. The transverse plane dissects the middle of the body between the chest and abdomen, separating the top of the body from the bottom. The midline is an imaginary line running through the middle of the body, from top to bottom. Figure 33.5 shown are the planes of a quadrupedal goat and a bipedal human. The midsagittal plane divides the body exactly in half, into right and left portions. The frontal plane divides the front and back, and the transverse plane divides the body into upper and lower portions. Vertebrate animals have a number of defined body cavities, as illustrated in Figure 33. 6. 2 of these are major cavities that contain smaller cavities within them. The dorsal cavity contains the cranial and the vertebral or spinal cavities. The ventral cavity contains the thoracic cavity, which in turn contains the pleural cavity around the lungs and the pericardial cavity, which surrounds the heart. The ventral cavity also contains the abdominopelvic cavity, which can be separated into the abdominal and the pelvic cavities. Illustration shows a cross-sectional side view of the upper part of a human body. The entire head region above the eyes and to the back of the head, and a long thin strip from this region down the back, is shaded to indicate the dorsal cavity. The head is labeled cranial cavity, and the long thin region down the back is the spinal cavity. A large oblong area shaded at the front of the body indicates the ventral cavity. It is labeled from top to bottom as thoracic cavity, diaphragm, thin lines separating regions, abdominal cavity, and pelvic cavity. The abdominal and pelvic cavities are separated by a thin dashed line, and together they are labeled the abdominopelvic cavity. Figure 33. 6 vertebrate animals have 2 major body cavities. The dorsal cavity contains the cranial and the spinal cavity. The ventral cavity contains the thoracic cavity and the abdominopelvic cavity. The thoracic cavity is separated from the abdominopelvic cavity by the diaphragm. The abdominopelvic cavity is separated into the abdominal cavity and the pelvic cavity by an imaginary line parallel to the pelvis bones. Credit: Modification of work by NCI. Career connection: Physical anthropologist. Physical anthropologists study the adaptation, variability, and evolution of human beings, plus their living and fossil relatives. They can work in a variety of settings, although most will have an academic appointment at a university, usually in an anthropology department or a biology, genetics, or zoology department. Non-academic positions are available in the automotive and aerospace industries, where the focus is on human size, shape, and anatomy. Research by these professionals might range from studies of how the human body reacts to car crashes to exploring how to make seats more comfortable. Other non-academic positions can be obtained in museums of natural history, anthropology, archaeology, or science and technology. These positions involve educating students from grade school through graduate school. Physical anthropologists serve as education coordinators, collection managers, writers for museum publications, and as administrators. Zoos employ these professionals, especially if they have an expertise in primate biology. They work in collection management and captive breeding programs for endangered species. Forensic science utilizes physical anthropology expertise in identifying human and animal remains, assisting in determining the cause of death, and for expert testimony in trials. Learning objectives. By the end of this section, you will be able to do the following. Describe epithelial tissues. Discuss the different types of connective tissues in animals. Describe 3 types of muscle tissues. Describe nervous tissue. The tissues of multicellular, complex animals are 4 primary types: epithelial, connective, muscle, and nervous. Recall that tissues are groups of similar cells, cells carrying out related functions. These tissues combine to form organs, like the skin or kidney, that have specific, specialized functions within the body. Organs are organized into organ systems to perform functions. Examples include the circulatory system, which consists of the heart and blood vessels, and the digestive system, consisting of several organs, including the stomach, intestines, liver, and pancreas. Organ systems come together to create an entire organism. Epithelial tissues. Epithelial tissues cover the outside of organs and structures in the body, and line the lumens of organs in a single layer or multiple layers of cells. The types of epithelia are classified by the shapes of cells present and the number of layers of cells. Epithelia composed of a single layer of cells is called simple epithelia. Epithelial tissue composed of multiple layers is called stratified epithelia. Table 33.2 summarizes the different types of epithelial tissues. Different types of epithelial tissues. Cell shape description location. Squamous flat, irregular round shape simple. Lung alveoli capillaries stratified. Skin, mouth, vagina. Cubital cube-shaped. Central nucleus glands, renal tubules. Columnar tall, narrow, nucleus toward base. Tall, narrow, nucleus along cell simple. Digestive tract, pseudostratified. Respiratory tract. Transitional round, simple but appear stratified urinary bladder. Table 33.2. Squamous epithelia. Squamous epithelial cells are generally round, flat, and have a small, centrally located nucleus. The cell outline is slightly irregular, and cells fit together to form a covering or lining. When the cells are arranged in a single layer, simple epithelia, they facilitate diffusion in tissues, such as the areas of gas exchange in the lungs and the exchange of nutrients and waste at blood capillaries. Illustration A shows irregularly shaped cells with a central nucleus. Micrograph B shows a cross-section of squamous cells from the human cervix. In the upper layer, the cells appear to be tightly packed. In the middle layer, they appear to be more loosely packed, and in the lower layer, they are flatter and elongated. Figure 33.7 squamous epithelia cells, A, have a slightly irregular shape and a small, centrally located nucleus. These cells can be stratified into layers, as in B, this human cervix specimen. Credit B, modification of work by Ed Uthman, scale bar data from Matt Russell. Figure 33.7a illustrates a layer of squamous cells with their membranes joined together to form an epithelium. Image Figure 33. 7b illustrates squamous epithelial cells arranged in stratified layers, where protection is needed on the body from outside abrasion and damage. This is called a stratified squamous epithelium and occurs in the skin and in tissues lining the mouth and vagina. Cubital epithelia. Cubital epithelial cells, shown in Figure 33.8, are cube-shaped with a single, central nucleus. They are most commonly found in a single layer representing a simple epithelia in glandular tissues throughout the body, where they prepare and secrete glandular material. They are also found in the walls of tubules and in the ducts of the kidney and liver. Illustration shows cells, shaped like slices of pie, arranged in a circle. The hub of the circle is empty. 3 of these circles of cells cluster together. Figure 33. 8 simple cubital epithelial cells line tubules in the mammalian kidney, where they are involved in filtering the blood. Columnar epithelia. Columnar epithelial cells are taller than they are wide. They resemble a stack of columns in an epithelial layer and are most commonly found in a single-layer arrangement. The nuclei of columnar epithelial cells in the digestive tract appear to be lined up at the base of the cells, as illustrated in Figure 33. 9. These cells absorb material from the lumen of the digestive tract and prepare it for entry into the body through the circulatory and lymphatic systems. Illustration shows tall, columnar cells arranged side by side. Each cell has a nucleus located near the bottom and cilia extending from the top. Two oval goblet cells are interspersed among the columnar epithelial cells. The goblet cells, which are shorter than the columnar cells, are in direct contact with the intestinal lumen. Beneath the columnar cells is a layer of horizontal cells. Figure 33. 9 simple columnar epithelial cells absorb material from the digestive tract. Goblet cells secrete mucus into the digestive tract lumen. Columnar epithelial cells lining the respiratory tract appear to be stratified. However, each cell is attached to the base membrane of the tissue, and therefore, they are simple tissues. The nuclei are arranged at different levels in the layer of cells, making it appear as though there is more than one layer, as seen in Figure 33. 10. This is called pseudostratified columnar epithelia. This cellular covering has cilia at the apical, or free, surface of the cells. The cilia enhance the movement of mucus and trapped particles out of the respiratory tract, helping to protect the system from invasive microorganisms and harmful material that has been breathed into the body. Goblet cells are interspersed in some tissues, such as the lining of the trachea. The goblet cells contain mucus that traps irritants, which in the case of the trachea, keep these irritants from getting into the lungs. Illustration shows columnar cells arranged side by side. The cells are wide at the top and thin at the bottom. Shorter columnar cells are interspersed between the lower, thin part of the tall columnar cells. Some of these cells extend to the surface of the epithelium, but they are very thin at the top. The nuclei of the tall columnar cells are located near the top, and the nuclei of the shorter columnar cells are located near the bottom, giving the appearance of two layers of cells. Cilia extend from the top of the tall columnar cells. Oval goblet cells are interspersed among the columnar epithelial cells. Beneath the columnar cells is a layer of horizontal cells. Figure 33. 10 pseudostratified columnar epithelia line the respiratory tract. They exist in one layer, but the arrangement of nuclei at different levels makes it appear that there is more than one layer. Goblet cells interspersed between the columnar epithelial cells secrete mucus into the respiratory tract. Transitional epithelia. Transitional or uroepithelial cells appear only in the urinary system, primarily in the bladder and ureter. These cells are arranged in a stratified layer, but they have the capability of appearing to pile up on top of each other in a relaxed, empty bladder, as illustrated in Figure 3311. As the urinary bladder fills, the epithelial layer unfolds and expands to hold the volume of urine introduced into it. As the bladder fills, it expands and the lining becomes thinner. In other words, the tissue transitions from thick to thin. Visual connection. Illustration shows tall, diamond-shaped cells layered one on top of the other. Figure 33. 11 transitional epithelia of the urinary bladder undergo changes in thickness depending on how full the bladder is. Which of the following statements about types of epithelial cells is false? Simple columnar epithelial cells line the tissue of the lung. Simple cuboidal epithelial cells are involved in the filtering of blood in the kidney. Pseudostratified columnar epithelia occur in a single layer, but the arrangement of nuclei makes it appear that more than one layer is present. Transitional epithelia change in thickness depending on how full the bladder is. Connective tissues. Connective tissues are made up of a matrix consisting of living cells and a non-living substance, called the ground substance. The ground substance is made of an organic substance, usually a protein, and an inorganic substance, usually a mineral or water. The principal cell of connective tissues is the fibroblast. This cell makes the fibers found in nearly all of the connective tissues. Fibroblasts are motile, able to carry out mitosis, and can synthesize whichever connective tissue is needed. Macrophages, lymphocytes, and occasionally, leukocytes can be found in some of the tissues. Some tissues have specialized cells that are not found in the others. Connective tissues are made up of living cells and a non-living matrix of organic substances, usually proteins, and inorganic substances, usually a mineral or water. The organic portion or protein fibers found in connective tissues are either collagen, elastic, or reticular fibers. Collagen fibers provide strength to the tissue, preventing it from being torn or separated from the surrounding tissues. Elastic fibers are made of the protein elastin. This fiber can stretch to 1½ of its length and return to its original size and shape. Elastic fibers provide flexibility to the tissues. Reticular fibers are the third type of protein fiber found in connective tissues. This fiber consists of thin strands of collagen that form a network of fibers to support the tissue and other organs to which it is connected. The various types of connective tissues, the types of cells and fibers they are made of, and sample locations of the tissues is summarized in Table 33.3. Connective tissues. Tissue cells fibers location. Loose/areolar fibroblasts. Macrophages, some lymphocytes, some neutrophils few. Collagen, elastic, reticular around blood vessels. Anchors epithelia. Dense, fibrous connective tissue fibroblasts. Macrophages mostly collagen irregular. Skin, regular. Tendons, ligaments. Cartilage chondrocytes, chondroblasts hyaline. Few, collagen fibrocartilage. Large amount of collagen shark skeleton, fetal bones, human ears, intervertebral discs. Bone osteoblasts, osteocytes, osteoclasts some. Collagen, elastic vertebrate skeletons. Adipose adipocytes few adipose, fat. Blood red blood cells, white blood cells none blood. Table 33.3. Loose areolar connective tissue. Loose connective tissue, also called areolar connective tissue, has a sampling of all of the components of a connective tissue. As illustrated in Figure 33.12, loose connective tissue has some fibroblasts. Macrophages are present as well. Collagen fibers are relatively wide and stain a light pink, while elastic fibers are thin and stain dark blue to black. The material in the connective tissue gives it a loose consistency similar to a cotton ball that has been pulled apart. Loose connective tissue is found around every blood vessel and helps to keep the vessel in place. The tissue is also found around and between most body organs. In summary, areolar tissue is tough, yet flexible, and comprises membranes. Illustration shows thick collagen fibers and thin elastin fibers loosely woven together in an irregular network. Oval fibroblasts are interspersed among the fibers. Figure 33. 12 loose connective tissue is composed of loosely woven collagen and elastic fibers. The fibers and other components of the connective tissue matrix are secreted by fibroblasts. Fibrous connective tissue. Fibrous connective tissues contain large amounts of collagen fibers and few cells or matrix material. The fibers can be arranged irregularly or regularly, with the strands lined up in parallel. Irregularly arranged fibrous connective tissues are found in areas of the body where stress occurs from all directions, such as the dermis of the skin. Regular fibrous connective tissue, shown in Figure 33.13, is found in tendons, which connect muscles to bones, and ligaments, which connect bones to bones. Illustration shows parallel collagen fibers packed tightly together. Interspersed among the collagen fibers are long, thin fibroblasts. Figure 33.13 fibrous connective tissue from the tendon has strands of collagen fibers lined up in parallel. Cartilage. Cartilage is a connective tissue with a large amount of the matrix of variable composition. The cells, called chondrocytes, make the matrix and fibers of the tissue. Chondrocytes are found in spaces within the tissue called lacunae. A cartilage with few collagen and elastic fibers is hyaline cartilage, illustrated in Figure 33.14. The lacunae are randomly scattered throughout the tissue, and the matrix takes on a milky or scrubbed appearance with routine histological stains. Sharks have cartilaginous skeletons, as does nearly the entire human skeleton during a specific pre-birth developmental stage. A remnant of this cartilage persists in the outer portion of the human nose. Hyaline cartilage is also found at the ends of long bones, reducing friction and cushioning the articulations of these bones. Illustration shows pairs of chondrocytes embedded in a matrix. The parts of the cells that face one another are flat, and the outer surfaces are rounded. Each cell has a small, rounded nucleus. Figure 33.14 14 hyaline cartilage consists of a matrix with cells called chondrocytes embedded in it. The chondrocytes exist in cavities in the matrix called lacunae. Elastic cartilage has a large amount of elastic fibers, giving it tremendous flexibility. The ears of most vertebrate animals contain this cartilage, as do portions of the larynx or voice box. Fibrocartilage contains a large amount of collagen fibers, giving the tissue tremendous strength. Fibrocartilage comprises the intervertebral discs in vertebrate animals. Hyaline cartilage found in movable joints, such as the knee and shoulder, becomes damaged as a result of age or trauma. Damaged hyaline cartilage is replaced by fibrocartilage and results in the joints becoming stiff. Bone, or osseous tissue, is a connective tissue that has a large amount of two different types of matrix material. The organic matrix is similar to the matrix material found in other connective tissues, including some amount of collagen and elastic fibers. This gives strength and flexibility to the tissue. The inorganic matrix consists of mineral salts, mostly calcium salts, that give the tissue hardness. Without adequate organic material in the matrix, the tissue breaks. Without adequate inorganic material in the matrix, the tissue bends. There are 3 types of cells in bone: osteoblasts, osteocytes, and osteoclasts. Osteoblasts are active in making bone for growth and remodeling. Osteoblasts deposit bone material into the matrix, and after the matrix surrounds them, they continue to live, but in a reduced metabolic state as osteocytes. Osteocytes are found in lacunae of the bone. Osteoclasts are active in breaking down bone for bone remodeling, and they provide access to calcium stored in tissues. Osteoclasts are usually found on the surface of the tissue. Bone can be divided into 2 types: compact and spongy. Compact bone is found in the shaft or diaphysis of a long bone and the surface of the flat bones, while spongy bone is found in the end or epiphysis of a long bone. Compact bone is organized into subunits called osteons, as illustrated in Figure 3315. A blood vessel and a nerve are found in the center of the structure within the Haversian canal, with radiating circles of lacunae around it, known as lamellae. The wavy lines seen between the lacunae are microchannels called canaliculi. They connect the lacunae to aid diffusion between the cells. Spongy bone is made of tiny plates called trabeculae. These plates serve as struts to give the spongy bone strength. Over time, these plates can break, causing the bone to become less resilient. Bone tissue forms the internal skeleton of vertebrate animals, providing structure to the animal and points of attachment for tendons. Illustration A shows a cross-section of a long bone with wide protrusions at either end. The outer part is compact bone. Inside the compact bone is porous spongy bone made of web-like trabeculae. The spongy bone fills the wide part at either end of the bone. In the middle, a hollow exists inside the spongy bone. Illustration B shows several circular osteons clustered together in compact bone. At the hub of each osteon is an opening called the Haversian canal, filled with blood and lymph vessels and nerves. The lamellae surrounding the Haversian canal resemble concentric circles. Lacunae are wide spaces that contain osteocytes in the rings between the lamellae. Microchannels called canaliculi radiate through the rings out from the central Haversian canal, connecting the lacunae together. Illustration C shows small osteoblasts surrounding the outside of bone. Larger osteoclasts are also on the outer surface, forming a hollow in the bone. Osteocytes are long, thin cells in the lacunae. Figure 33. 15. A compact bone is a dense matrix on the outer surface of bone. Spongy bone, inside the compact bone, is porous with web-like trabeculae. B. Compact bone is organized into rings called osteons. Blood vessels, nerves, and lymphatic vessels are found in the central Haversian canal. Rings of lamellae surround the Haversian canal. Between the lamellae are cavities called lacunae. Canaliculi are microchannels connecting the lacunae together. C. Osteoblasts surround the exterior of the bone. Osteoclasts bore tunnels into the bone, and osteocytes are found in the lacunae. Adipose tissue, or fat tissue, is considered a connective tissue even though it does not have fibroblasts or a real matrix. Adipose tissue is made up of cells called adipocytes that collect and store fat in the form of triglycerides, for energy metabolism. Adipose tissues additionally serve as insulation to help maintain body temperatures, allowing animals to be endothermic, and they function as cushioning against damage to body organs. Under a microscope, adipose tissue cells appear empty due to the extraction of fat during the processing of the material for viewing, as seen in Figure 33. 16. The thin lines in the image are the cell membranes, and the nuclei are the small, black dots at the edges of the cells. Illustration shows irregularly shaped cells with tiny nuclei clustered next to the cell's outer membrane. Figure 33. 16. Adipose is a connective tissue made up of cells called adipocytes. Adipocytes have small nuclei localized at the cell edge. Blood is considered a connective tissue because it has a matrix, as shown in Figure 33. 17. The living cell types are red blood cells, RBC, also called erythrocytes, and white blood cells, WBC, also called leukocytes. The fluid portion of whole blood, its matrix, is commonly called plasma. Different types of blood cells are shown. Red blood cells are disc-shaped, with a central indentation. Platelets are much smaller than red blood cells, narrow and long. Neutrophils, eosinophils, lymphocytes, monocytes, and basophils are all about 3 times the diameter of a red blood cell and round. They differ in the shape of the nucleus and in the presence or absence of granules in the cytoplasm. Macrophages, which are the largest cell type, have pseudopods which give them an irregular shape. Figure 33. 17. Blood is a connective tissue that has a fluid matrix, called plasma, and no fibers. Erythrocytes, red blood cells, the predominant cell type, are involved in the transport of oxygen and carbon dioxide. Also present are various leukocytes, white blood cells, involved in immune response. The cell found in greatest abundance in blood is the erythrocyte. Erythrocytes are counted in millions in a blood sample. The average number of red blood cells in primates is 47 to 55 million cells per microliter. Erythrocytes are consistently the same size in a species, but vary in size between species. For example, the average diameter of a primate red blood cell is 7.5 microM. A dog is close at 7.0 microM, but a cat's RBC diameter is 5.9 microM. Sheep erythrocytes are even smaller at 4.6 microM. Mammalian erythrocytes lose their nuclei and mitochondria when they are released from the bone marrow where they are made. Fish, amphibian, and avian red blood cells maintain their nuclei and mitochondria throughout the cell's life. The principal job of an erythrocyte is to carry and deliver oxygen to the tissues. Leukocytes are the predominant white blood cells found in the peripheral blood. Leukocytes are counted in the thousands in the blood, with measurements expressed as ranges. Primate counts range from 4,800 to 10,800 cells per microL. Dogs from 5,600 to 19,200 cells per microL. Cats from 8,000 to 25,000 cells per microL. Cattle from 4,000 to 12,000 cells per microL, and pigs from 11,000 to 22,000 cells per microL. Lymphocytes function primarily in the immune response to foreign antigens or material. Different types of lymphocytes make antibodies tailored to the foreign antigens and control the production of those antibodies. Neutrophils are phagocytic cells, and they participate in one of the early lines of defense against microbial invaders, aiding in the removal of bacteria that have entered the body. Another leukocyte that is found in the peripheral blood is the monocyte. Monocytes give rise to phagocytic macrophages that clean up dead and damaged cells in the body, whether they are foreign or from the host animal. 2 additional leukocytes in the blood are eosinophils and basophils. Both help to facilitate the inflammatory response. The slightly granular material among the cells is a cytoplasmic fragment of a cell in the bone marrow. This is called a platelet or thrombocyte. Platelets participate in the stages leading up to coagulation of the blood to stop bleeding through damaged blood vessels. Blood has a number of functions, but primarily it transports material through the body to bring nutrients to cells and remove waste material from them. Muscle tissues. There are 3 types of muscle in animal bodies: smooth, skeletal, and cardiac. They differ by the presence or absence of striations or bands, the number and location of nuclei, whether they are voluntarily or involuntarily controlled, and their location within the body. Table 33. 4 summarizes these differences. Types of muscles, type of muscle striations, nuclei control location, smooth, no single, in center involuntary visceral organ skeletal, yes many, at periphery voluntary skeletal muscles, cardiac yes single, in center involuntary heart, table 33. 4 smooth muscle does not have striations in its cells. It has a single, centrally located nucleus, as shown in figure 3318. Constriction of smooth muscle occurs under involuntary, autonomic nervous control and in response to local conditions in the tissues. Smooth muscle tissue is also called non-striated, as it lacks the banded appearance of skeletal and cardiac muscle. The walls of blood vessels, the tubes of the digestive system, and the tubes of the reproductive systems are composed of mostly smooth muscle. The smooth muscle cells are long and arranged in parallel bands. Each cell has a long, narrow nucleus. Skeletal muscle cells are also long but have striations across them and many small nuclei per cell. Cardiac muscles are shorter than smooth or skeletal muscle cells, and each cell has one nucleus. Figure 33. 18 smooth muscle cells do not have striations, while skeletal muscle cells do. Cardiac muscle cells have striations, but unlike the multinucleate skeletal cells, they have only one nucleus. Cardiac muscle tissue also has intercalated discs, specialized regions running along the plasma membrane that join adjacent cardiac muscle cells and assist in passing an electrical impulse from cell to cell. Skeletal muscle has striations across its cells caused by the arrangement of the contractile proteins actin and myosin. These muscle cells are relatively long and have multiple nuclei along the edge of the cell. Skeletal muscle is under voluntary somatic nervous system control and is found in the muscles that move bones. Figure 33. 18 illustrates the histology of skeletal muscle. Cardiac muscle, shown in figure 3318, is found only in the heart. Like skeletal muscle, it has cross striations in its cells, but cardiac muscle has a single, centrally located nucleus. Cardiac muscle is not under voluntary control, but can be influenced by the autonomic nervous system to speed up or slow down. An added feature to cardiac muscle cells is a line that extends along the end of the cell as it abuts the next cardiac cell in the row. This line is called an intercalated disc. It assists in passing electrical impulse efficiently from one cell to the next and maintains the strong connection between neighboring cardiac cells. Nervous tissues are made of cells specialized to receive and transmit electrical impulses from specific areas of the body, and to send them to specific locations in the body. The main cell of the nervous system is the neuron, illustrated in figure 33. 19. The large structure with a central nucleus is the cell body of the neuron. Projections from the cell body are either dendrites specialized in receiving input or a single axon specialized in transmitting impulses. Some glial cells are also shown. Astrocytes regulate the chemical environment of the nerve cell, and oligodendrocytes insulate the axon so the electrical nerve impulse is transferred more efficiently. Other glial cells that are not shown support the nutritional and waste requirements of the neuron. Some of the glial cells are phagocytic and remove debris or damaged cells from the tissue. A nerve consists of neurons and glial cells. Illustration shows a neuron which has an oval cell body, labeled as a soma. Branch-like dendrites extend from 3 sides of the body. A long, thin axon extends from the 4th side. At the end of the axon are branch-like terminals, called axon terminals. A cell called an oligodendrocyte grows alongside the axon. Projections from the oligodendrocyte wrap around the axon, forming a myelin sheath. Another cell called an astrocyte sits alongside the axon. Figure 33. 19. The neuron has projections called dendrites that receive signals and projections called axons that send signals. Also shown are 2 types of glial cells. Astrocytes regulate the chemical environment of the nerve cell, and oligodendrocytes insulate the axon so the electrical nerve impulse is transferred more efficiently. Career connection pathologist. A pathologist is a medical doctor or veterinarian who has specialized in the laboratory detection of disease in animals, including humans. These professionals complete medical school education and follow it with an extensive postgraduate residency at a medical center. A pathologist may oversee clinical laboratories for the evaluation of body tissue and blood samples for the detection of disease or infection. They examine tissue specimens through a microscope to identify cancers and other diseases. Some pathologists perform autopsies to determine the cause of death and the progression of disease. Learning objectives by the end of this section, you will be able to do the following. Define homeostasis, describe the factors affecting homeostasis, discuss positive and negative feedback mechanisms used in homeostasis, describe thermoregulation of endothermic and ectothermic animals, animal organs and organ systems constantly adjust to internal and external changes through a process called homeostasis. Steady state. These changes might be in the level of glucose or calcium in blood or in external temperatures. Homeostasis means to maintain dynamic equilibrium in the body. It is dynamic because it is constantly adjusting to the changes that the body's systems encounter. It is equilibrium because body functions are kept within specific ranges. Even an animal that is apparently inactive is maintaining this homeostatic equilibrium. Homeostatic process, the goal of homeostasis, is the maintenance of equilibrium around a point or value called a set point. While there are normal fluctuations from the set point, the body's systems will usually attempt to go back to this point. A change in the internal or external environment is called a stimulus and is detected by a receptor. The response of the system is to adjust the deviation parameter toward the set point. For instance, if the body becomes too warm, adjustments are made to cool the animal. If the blood's glucose rises after a meal, adjustments are made to lower the blood glucose level by getting the nutrient into tissues that need it or to store it for later use. Control of homeostasis. When a change occurs in an animal's environment, an adjustment must be made. The receptor senses the change in the environment, then sends a signal to the control center. In most cases, the brain, which in turn generates a response that is signaled to an effector. The effector is a muscle that contracts or relaxes, or a gland that secretes. Homeostasis is maintained by negative feedback loops. Positive feedback loops actually push the organism further out of homeostasis, but may be necessary for life to occur. Homeostasis is controlled by the nervous and endocrine system of mammals. Negative feedback mechanisms. Any homeostatic process that changes the direction of the stimulus is a negative feedback loop. It may either increase or decrease the stimulus, but the stimulus is not allowed to continue as it did before the receptor sensed it. In other words, if a level is too high, the body does something to bring it down, and conversely, if a level is too low, the body does something to make it go up. Hence the term negative feedback. An example is animal maintenance of blood glucose levels. When an animal has eaten, blood glucose levels rise. This is sensed by the nervous system. Specialized cells in the pancreas sense this, and the hormone insulin is released by the endocrine system. Insulin causes blood glucose levels to decrease, as would be expected in a negative feedback system, as illustrated in Figure 33.20. However, if an animal has not eaten and blood glucose levels decrease, this is sensed in another group of cells in the pancreas, and the hormone glucagon is released, causing glucose levels to increase. This is still a negative feedback loop, but not in the direction expected by the use of the term negative. Another example of an increase as a result of the feedback loop is the control of blood calcium. If calcium levels decrease, specialized cells in the parathyroid gland sense this and release parathyroid hormone, PTH, causing an increased absorption of calcium through the intestines and kidneys, and possibly the breakdown of bone in order to liberate calcium. The effects of PTH are to raise blood levels of the element. Negative feedback loops are the predominant mechanism used in homeostasis. Illustration shows the response to consuming a meal. When food is consumed and digested, blood glucose levels rise. In response to the higher concentration of glucose, the pancreas secretes insulin into the blood. In response to the higher insulin levels in the blood, glucose is transported into many body cells. Liver cells store glucose as glycogen. As a result, glucose levels drop. In response to the lower concentration of glucose, the pancreas stops secreting insulin. Figure 33. 20 blood sugar levels are controlled by a negative feedback loop. Credit. Modification of work by John Sullivan, Positive feedback loop. A positive feedback loop maintains the direction of the stimulus, possibly accelerating it. Few examples of positive feedback loops exist in animal bodies, but one is found in the cascade of chemical reactions that result in blood clotting or coagulation. As one clotting factor is activated, it activates the next factor in sequence until a fibrin clot is achieved. The final factor, a protein called thrombin, converts inactive fibrinogen to fibrin. Thrombin also activates additional clotting factor molecules earlier in the cascade, resulting in the activation of even more thrombin. This positive feedback loop amplifies the cascade for faster, more effective formation of a blood clot. Another example of positive feedback is uterine contractions during childbirth, as illustrated in Figure 3321. The hormone oxytocin, made by the endocrine system, stimulates the contraction of the uterus. This stimulation is sensed by the nervous system. Instead of lowering the oxytocin, more oxytocin is produced until the contractions are powerful enough to produce childbirth. Visual connection prior to birth, the baby pushes against the cervix, causing it to stretch. Stretching of the cervix causes nerve impulses to be sent to the brain. As a result, the brain stimulates the pituitary to release oxytocin. Oxytocin causes the uterus to contract. As a result, the baby pushes against the cervix in a positive feedback loop. Figure 33. 21. The birth of a human infant is the result of positive feedback. State whether each of the following processes is regulated by a positive feedback loop or a negative feedback loop. A person feels satiated after eating a large meal. The blood has plenty of red blood cells. As a result, erythropoietin, a hormone that stimulates the production of new red blood cells, is no longer released from the kidney. Set point: it is possible to adjust a system's set point. When this happens, the feedback loop works to maintain the new setting. An example of this is blood pressure. Over time, the normal or set point for blood pressure can increase as a result of continued increases in blood pressure. The body no longer recognizes the elevation as abnormal, and no attempt is made to return to the lower set point. The result is the maintenance of an elevated blood pressure that can have harmful effects on the body. Medication can lower blood pressure and lower the set point in the system to a more healthy level. This is called a process of alteration of the set point in a feedback loop. Changes can be made in a group of body organ systems in order to maintain a set point in another system. This is called acclimatization. This occurs, for instance, when an animal migrates to a higher altitude than that to which it is accustomed. In order to adjust to the lower oxygen levels at the new altitude, the body increases the number of red blood cells circulating in the blood to ensure adequate oxygen delivery to the tissues. Another example of acclimatization is animals that have seasonal changes in their coats. A heavier coat in the winter ensures adequate heat retention, and a light coat in summer assists in keeping body temperature from rising to harmful levels. Link to learning feedback mechanisms can be understood in terms of driving a race car along a track. Watch a short video lesson on positive and negative feedback loops. Homeostasis. Thermoregulation: body temperature affects body activities. Generally, as body temperature rises, enzyme activity rises as well. For every 10 degrees centigrade rise in temperature, enzyme activity doubles, up to a point. Body proteins, including enzymes, begin to denature and lose their function with high heat, around 50 OC for mammals. Enzyme activity will decrease by half for every 10 degrees centigrade drop in temperature, to the point of freezing, with a few exceptions. Some fish can withstand freezing solid and return to normal with thawing. Link to learning: watch this video on thermoregulation to see illustrations of this process in a variety of animals. Endotherms and ectotherms animals can be divided into two groups. Some maintain a constant body temperature in the face of differing environmental temperatures, while others have a body temperature that is the same as their environment and thus varies with the environment. Animals that rely on external temperatures to set their body temperature are ectotherms. This group has been called cold-blooded, but the term may not apply to an animal in the desert with a very warm body temperature. In contrast to ectotherms, poikilotherms are animals with constantly varying internal temperatures. An animal that maintains a constant body temperature in the face of environmental changes is called a homeotherm. Endotherms are animals that rely on internal sources for maintenance of relatively constant body temperature in varying environmental temperatures. These animals are able to maintain a level of metabolic activity in cooler external temperatures, which an ectotherm cannot do to differing enzyme levels of activity. It is worth mentioning that some ectotherms and poikilotherms have relatively constant body temperatures due to the constant environmental temperatures in their habitats. These animals are so-called ectothermic homeotherms, like some deep-sea fish species. Heat can be exchanged between an animal and its environment through 4 mechanisms: radiation, evaporation, convection, and conduction, figure 3322. Radiation is the emission of electromagnetic heat waves. Heat comes from the sun in this manner and radiates from dry skin the same way. Heat can be removed with liquid from a surface during evaporation. This occurs when a mammal sweats. Convection currents of air remove heat from the surface of dry skin as the air passes over it. Heat will be conducted from one surface to another during direct contact with the surfaces, such as an animal resting on a warm rock. Photo A shows the sun. Photo B shows a sweaty person. Photo C shows a lion with its mane blowing in the wind. Photo D shows a person holding a steaming hot drink. Figure 33. 22 heat can be exchanged by 4 mechanisms: A, radiation, B, evaporation, C, convection, or D, conduction. Credit B, modification of work by Culles/Flicker. Credit C, modification of work by Chad Rosenthal. Credit D, modification of work by Stacy. D/Flicker. Heat conservation and dissipation. Animals conserve or dissipate heat in a variety of ways. In certain climates, endothermic animals have some form of insulation, such as fur, fat, feathers, or some combination thereof. Thick fur and feathers trap a layer of air that helps to provide thermal insulation. Polar bears and seals live and swim in a subfreezing environment and yet maintain a constant, warm body temperature. The Arctic fox, for example, uses its fluffy tail as extra insulation when it curls up to sleep in cold weather. Mammals have a residual effect from shivering and increased muscle activity. Erector pili muscles cause goosebumps, causing small hairs to stand up when the individual is cold. This has the intended effect of increasing body temperature. Mammals use layers of fat to achieve the same end. Loss of significant amounts of body fat will compromise an individual's ability to conserve heat. Endotherms use their circulatory systems to help maintain body temperature. Vasodilation brings more blood and heat to the body surface, facilitating radiation and evaporative heat loss, which helps to cool the body. Vasoconstriction reduces blood flow in peripheral blood vessels, forcing blood toward the core and the vital organs found there, and conserving heat. Some animals have adaptations to their circulatory system that enable them to transfer heat from arteries to veins, warming blood returning to the heart. This is called a countercurrent heat exchange. It prevents the cold venous blood from cooling the heart and other internal organs. This adaptation can be shut down in some animals to prevent overheating the internal organs. The countercurrent adaptation is found in many animals, including dolphins, sharks, bony fish, bees, and hummingbirds. In contrast, similar adaptations can help cool endotherms when needed, such as dolphin flukes and elephant ears. Some ectothermic animals use changes in their behavior to help regulate body temperature. For example, a desert ectothermic animal may simply seek cooler areas during the hottest part of the day in the desert to keep from getting too warm. The same animals may climb onto rocks to capture heat during a cold desert night. Some animals seek water to aid evaporation in cooling them, as seen with reptiles. Other ectotherms use group activity, such as the activity of bees, to warm a hive to survive winter. Many animals, especially mammals, use metabolic waste heat as a heat source. When muscles are contracted, Most of the energy from the ATP used in muscle actions is wasted energy that translates into heat. Severe cold elicits a shivering reflex that generates heat for the body. Many species also have a type of adipose tissue called brown fat that specializes in generating heat. Neural control of thermoregulation. The nervous system is important to thermoregulation, as illustrated in Figure 33. 22. The processes of homeostasis and temperature control are centered in the hypothalamus of the advanced animal brain. Visual connection flow chart shows how normal body temperature is maintained. If the body temperature rises, blood vessels dilate, resulting in loss of heat to the environment. Sweat glands secrete fluid. As this fluid evaporates, heat is lost from the body. As a result, the body temperature falls to normal body temperature. If body temperature falls, blood vessels constrict so that heat is conserved. Sweat glands do not secrete fluid. Shivering, involuntary contraction of muscles, releases heat which warms the body. Heat is retained, and body temperature increases to normal. Figure 33. 23. The body is able to regulate temperature in response to signals from the nervous system. When bacteria are destroyed by leukocytes, pyrogens are released into the blood. Pyrogens reset the body's thermostat to a higher temperature, resulting in fever. How might pyrogens cause the body temperature to rise? The hypothalamus maintains the set point for body temperature through reflexes that cause vasodilation and sweating when the body is too warm, or vasoconstriction and shivering when the body is too cold. It responds to chemicals from the body. When a bacterium is destroyed by phagocytic leukocytes, chemicals called endogenous pyrogens are released into the blood. These pyrogens circulate to the hypothalamus and reset the thermostat. This allows the body's temperature to increase in what is commonly called a fever. An increase in body temperature causes iron to be conserved, which reduces a nutrient needed by bacteria. An increase in body heat also increases the activity of the animal's enzymes and protective cells while inhibiting the enzymes and activity of the invading microorganisms. Finally, heat itself may also kill the pathogen. A fever that was once thought to be a complication of an infection is now understood to be a normal defense mechanism.